Jai Hind and welcome to all. Myself Shiwani Singhal from Ajay Kumar Garg Engineering College, Ghaziabad, Department of Applied Science and Humanities will deliver a lecture on spectroscopic techniques and application. This is a part of Unit 2 and this will be my first lecture where I will discuss the following topics: electromagnetic radiation and its energy, what is the electromagnetic spectrum and the types of spectroscopy which are mentioned in AKTU syllabus, and colorimetry or spectrophotometer. And to prepare these slides, I have taken the following references. And first, the elementary organic spectroscopy by Y.R. Sharma, engineering chemistry by Shashi Chawla, by Jain and Jain, and industrial methods of chemical analysis by B.K. Sharma. So, this is the first lecture where I will discuss what is the spectroscopy. So, spectroscopy means where we discuss how much light is scattered, absorbed or emit, or we can say transmitted when it passes through the medium. So, we can say that it is the branch of science where we study the interaction of matter with electromagnetic radiation. That is, we take the sample and irradiate with electromagnetic radiation of particular frequency, and then what type of transition occur we read in this spectroscopy. And this spectroscopy is very useful because in this case we do not require a sample in a large amount and we can reuse the sample, and this is fully automatic. So, and we know that the molecules had very small dimensions. So, it is very difficult to find the structure or read all these things. So, through spectroscopy we can study the overall structure of the molecule. So, the most consequences of such interaction is that energy is absorbed or emitted by the matter in discrete amount, which is called quanta. It is one of the most powerful tools for the study of atomic or molecular structure, and it is also used in analysis of wide range of samples. So, before all this spectroscopy, first we discuss what is the spectrum and what is the origin of a spectrum. So, to discuss this or to understand this, we know that basically there are various energy level in an atom or molecule. The lower energy level is called ground state energy level, which is shown by Eg, and higher level, these are known as excited energy level, which are shown by Ee. So, Eg, that is ground energy level, Ee, that is the upper or higher energy state energy level. So, when the molecule, atom or electron jump from or goes from ground state to higher energy state, it means it will absorb the energy and which is equal to h nu, and which arise or which appear as a absorption spectrum. So, simple, it results when the electron or atom or molecule goes from lower to higher energy state by the absorption of photon of energy, which is h nu. And here when the absorption occur, we get this type of absorption spectra, that is in a band of colorful line, or and we know that when we pass the white light from the prism, we get the 7 color spectrum. So, this is the 7 color spectrum, but at particular frequency where the absorption occur, we get as a dark line. And when the electron jump from higher state to lower energy state, it means it will emit the energy, and which is equal to h nu, and here we get emission spectrum. Emission spectrum means the emitted line, it will appear as a colored line and in a band of dark color. So, this is the type of absorption spectrum and this is the type of emission spectrum. Now, spectroscopic techniques. There are large or various type of spectroscopic techniques, such like electronic spectroscopy and magnetic spectroscopy, but here we read or we discuss only atomic spectroscopy and molecular spectroscopy. And on the basis of emission or absorption, we may say the absorption spectroscopy or emission spectroscopy. In atomic spectroscopy, here we study the interaction of electron or transition of electron from in between electronic energy level. In molecular spectroscopy, here we discuss the overall transition in a molecule. It means in atomic spectroscopy we discuss only electronic transition, but we know that instead of this electronic transition, there are other transitions between the energy level, and these energy levels are called vibrational energy level, rotational energy level. It means when we study the overall transition in between the energy level in a molecule, it gives the information about the molecular structure, and which is molecular spectroscopy. And where we discuss or read only electronic transition, it comes in electronic spectroscopy. So, although it is molecular spectroscopy is comparatively more complicated, but it gives a wide range of information. So, what is the basic theory? Suppose this is a matter or sample, and when we pass the incident light, and suppose intensity I naught, then we know that few part of this light it get reflect, which is in points very less, and few part of this it get absorb and transmit. So, absorption or transmission, it gives the information about these three spectroscopy, which is UV visible, IR and rotational or microwave spectroscopy. It means in this spectroscopy we relate how much light get absorbed or transmit when it pass through the medium or the matter. But few part means suppose 0.001th part of this light it may get scattered, and the concept scattering we use this in Raman spectroscopy. And third is photoluminescence, it is totally different, which comes in photochemistry. That is, we can say the thing it can emit the photon and it shines, and here phosphorescence, fluorescence it comes, but in this spectroscopy we consider only these two parts, that is how much light is scattered and how much light is absorbed. So, we talk about the light. So, first we should know that what is the EMR, that is electromagnetic radiation. We know that the light has a particle nature, but it could not explain the various phenomena like diffraction, refraction or etc. So, to explain this, it was discussed that the light has also wave nature. So, the radiant energy has wave nature and it is associated with electric field as well as magnetic field, and this type of radiations are called electromagnetic radiation. So, electromagnetic radiation is a form of energy that is transmitted through the space at an enormous velocity, and electromagnetic radiation has electric vector as well as magnetic vector, and both are coplanar, that is both are perpendicular. So, this is, suppose this is the electric vector and magnetic vector is like this is shown by the blue portion, and both are perpendicular to each other. So, we can say that EMR is the alternating electrical and associated magnetic force field in space, and thus it has electric component as well as magnetic component, and these two components oscillate in plane perpendicular to each other and which are in the direction of the propagation of the radiation. And the velocity of EMR in vacuum is independent of frequency, and which has the value 3 into 10 to the power 8 meter per second. In initial calculation of spectroscopy, the unit plays an important role. So, be aware, if you take in centimeter, then all units should be shown in centimeter, centimeter inverse or etc. So, aware about the units. Now, there are few terms which are associated with electromagnetic radiation, and we took the wave nature, suppose this. So, first term is wavelength. Wavelength, the distance between two successive crest or through of a wave is called wavelength. That is the distance between these two or the distance between these two, it is called wavelength, and which is shown by lambda, and the unit of lambda is meter, centimeter, it may be in angstrom, picometer, etc. And we know that 1 angstrom is equal to 10 to the power minus 10 meter or 10 to the power minus 8 centimeter, this, and picometer is 10 to the power minus 12 meter or 10 to the power minus 10 centimeter. Now, next is frequency. We know that the frequency is the number of cycle per second. So, here we can say that the number of complete wavelength unit passing through a given point in a unit time, it may be in 1 second or etc. And this frequency is shown by nu, and nu is equal to c upon lambda, that is the wave number, sorry, that is the wavelength and frequency, both are reciprocal to each other. So, suppose I am saying this is the increasing order of wavelength, so automatically it will be the decreasing order of frequency. And third is wave number. Wave number is the number of waves per second and which is equal to 1 upon lambda. So, this is the very good term which may be directly proportional to energy, because the energy, it is equal to h nu, and h nu is equal to h c upon lambda, and we can write this h c nu bar. So, nu wave number, it is shown by nu bar. Now, energy. So, we can say that the photons are quantized, and the Planck suggests that the radiation, it comes in quantized packets and which is equal to E is equal to h nu. So, this is the first simple numerical in spectroscopy. Suppose the frequency is given and examiner asks what is the energy of this radiation. So, we can apply direct formula E is equal to h nu, and suppose lambda is given, then we can apply this formula E is equal to h c upon lambda, where h is equal to Planck constant and nu is equal to frequency, and unit of this frequency is time inverse and generally in second inverse. And the Planck constant value is fixed, which is equal to 6.626 into 10 to the power minus 34 joule second. So, there are three conversion. First, E is equal to h nu, put the value of nu which is equal to c upon lambda, and 1 upon lambda is wave number. So, we can write E is equal to h c nu bar. So, first thing, lambda inversely proportional to frequency, and frequency is directly proportional to energy. That is, we can say, suppose here write this. So, suppose I know the order of frequency, we can correlate direct this relation with energy. Now, this is the simple rough diagram of a spectrophotometer, and a spectrophotometer means find the relation how much light get absorbed and when it pass through the medium. Suppose the intensity of incident light is I naught, and after passing through the medium its intensity become I. So, the relation between I and I naught we can find through spectrophotometer. This is the light source, and when we pass through the monochromator, and when after this sample we get the ratio or the how much light get scattered with respect to initial light, and we get this type of graph when we plot between absorbance means how much light get absorbed with wave number, or we can say with frequency. Now, next is electromagnetic spectrum. The arrangement of electromagnetic radiation in order of their increasing wavelength or decreasing frequency, because both are inversely proportional. So, the spectrum which is obtained is called electromagnetic spectrum. This is the order towards in decreasing wavelength. It means the radio waves has highest wavelength, and the cosmic gamma X-rays, these has least wavelength. So, this is the decreasing order of wavelength, and automatically we can say this is the increasing order of frequency. So, radio waves has least frequency while these have highest frequency. So, radio waves, microwaves, IR, then visible UV, X-rays, gamma rays and cosmic rays. Now, this is the comparative chart. radio waves have frequency in order 10 to the power 6 to 10 to the power 10, and while X-rays has 10 to the power 18 to 20. So, it increases. Wavelength, it is about 10 meter to 1 centimeter order, and X-rays, gamma rays about in nanometer or picometer. It means the wavelength decreases. Now, the next question is, there are lots of spectroscopy, and we know that the molecule there is the distribution of energy level in a molecule. So, which type of radiation is required for which type of spectroscopy? So, to find this, let we take this diagram, but before this diagram we should know how much type of energy are associated in a molecule. First is electronic energy level. Electronic energy level is associated with transition of electron from ground state to higher excited state, that is in molecular orbitals. Second is in vibrational energy. It means in a molecule, if the center of gravity does not change, but it does to and fro motion like upon pendulum, like a spring around the nuclei of molecule, then the molecule is said to have vibrational energy. Third is rotational energy. Rotational energy means if we imagine an axis perpendicular to the center of gravity and the molecule rotate along this axis. So, this is it means the molecule it is said to be that molecule has rotational energy. Now, translational energy means during the motion, if the center of gravity of molecule changes, then the molecule possesses translational energy. So, the total energy with the molecule associated, E is equal to electronic energy plus vibrational plus rotational plus translational. And since the translational energy is very less as compared to this, so it can be neglected. So, Born-Oppenheimer approximation, so we can say that the total energy associated with the molecule is the sum of electronic energy transition plus vibrational plus rotational. So, this is the basically distribution or energy level. This is the ground state and this is the excited state. And in ground state we see that there are vibrational energy level as well as in excited state. And these vibrational energy level are shown by V. Suppose V double dash, this is a simple notation and it is shown by V dash. So, it is shown by V. And these vibrational energy level, these are associated with groups of energy level which are called rotational energy level, that is J. So, simple if we can compare or we can explain by this example. Suppose we take two floor, ground floor and first floor, and in ground floor there are 9 rooms and in one room there are 20 seats. So, we can say that the 20 seats are comparatively very near. So, this is the group of rotational energy level and 20 seats are in one room, it means vibrational energy level, and 9 rooms are in one level and 9 rooms are in another level. So, these are ground state or excited state. Is it clear? So, ground state, excited state compare with floor, vibrational energy level with compare with the rooms, and rotational energy level means a seats present in rooms. So, here we see that for rotation it means the transition from J to 0, 1 to 2, 2 to 3, a very less amount of energy is required, and very less amount of energy is required means if we compare with this, the microwave, it has lower or less frequency, it means it has less energy, that is for rotation microwaves are sufficient. It means for rotational spectroscopy microwaves are sufficient, that is we pass or we irradiate with microwaves, and in this the transition between rotational energy level occur. Now, if we discuss or consider the transition in vibrational energy level, and for this transition comparatively high energy is required than rotation, but less energy is required for in transition. It means if we compare the chart, infrared, it has higher frequency than microwave, but lesser frequency than UV visible. That is, for vibrations, infrareds are sufficient. It means when we talk about the IR spectroscopy, so here we discuss or we took the point vibrational transitions. Now, third is transition between state, that is ground state to higher energy state. Here comparatively large amount of energy is required, and large amount of energy is required means if we compare these three, UV visible has the highest energy as compared to these two. It means for transition we took or we irradiate with UV visible spectroscopy. Okay. So, for rotational, microwaves are sufficient, for vibrations infrareds are sufficient, and for electronic transitions UV visible radiations are sufficient. Now, we compare 4 radiation spectroscopy, and there are other spectroscopy, but in AKTU syllabus these 4 spectroscopies are mentioned. So, we will discuss only these 4. So, first is rotational. Rotational means the transition between rotational level, and for rotations microwaves are sufficient, and microwaves has energy or wave number in order 1 to 100 centimeter inverse. And what is the condition? It is not necessary that all molecules will give rotational spectra. There is the limited condition. So, for rotational to get the rotational spectra or for rotation, there is a condition that molecule must have permanent dipole moment. This is the basically rough glance here, and it is an introduction part where we can find or get some idea then what type of points we discuss in detail. So, criteria or condition is it must have permanent dipole moment. For example, H2. So, H2 here we dipole moment mu, which is the charge separation, the distance into Q into D. So, H2 it has dipole moment 0. It means it will not give rotational spectra. And what is the use of this rotational spectroscopy? It is very useful to find the bond length of diatomic molecule. Second, vibrational or vibrational rotational energy level. So, here the transition between vibrational energy level occur, and for this vibrations IR radiations are sufficient, and the range is 500 to 4000 centimeter inverse. We will read this in detail in further lectures. And the criteria is during the vibration the dipole moment must change. For example, in CO2, if the vibration is symmetric, it means we stretch in symmetrical manner. In this case, the dipole moment will not change. That is, this type of vibration will not give IR vibrational or IR spectra. But if the stretching is asymmetrical, asymmetrical means not equal. In this condition, the dipole moment get changed. It means asymmetrical vibration or asymmetrical stretching will provide or will give vibrational spectra. Okay. So, this is the criteria or condition. So, in those molecules where the dipole moment get changed during vibration will show IR spectra. And what is the main application? The main application, it shows or it gives the information of functional group and the overall structure of the molecule. Third, and these three are absorption basically spectroscopy, and while fourth is scattering. So, for third, UV electronic spectroscopy. So, here the transition in between electronic energy level occur, and for this we radiate with UV visible, and that is why it is also known as UV visible spectroscopy. The range is visible as well as UV. Visible is, if we took in a wavelength, it is about 3800 to 7800, that is about 8000 angstrom, or if we took in frequency, then 12500 to 35000 per centimeter and 25000 to 70000 centimeter inverse. And what is the criteria? The criteria is the either conjugation means alternate double bond. So, the transition will occur, or chromophore must be present. Chromophore means colored group. And what is the main information which we get? The presence of conjugated double bond. So, this spectroscopy is very useful to find the conjugation. Suppose in a structure of vitamins, we can find how much, which type of conjugation is present, how much double bond is present, either the double bond is attached with direct with ring, we can find with the help of this spectroscopy. And fourth is Raman spectroscopy, and which is found by scattered light. So, here the transition between vibrational and rotational level occur, and this occur for this, the region is far and near IR region, but nowadays visible light, with a visible light we can find this type of a spectrum. And what is the criteria? The criteria is there must be a periodic change in polarizability during the vibration or rotation. And polarizability we find, suppose in case of H2, we know that there is a equal distribution, but when we applied external field, then definitely polarizability occur. So, there must be a periodic change in polarizability. We will discuss in detail in lecture, especially in Raman spectroscopy. And the information which from which we get is identify the light scattered properties. Okay. So, these 4 spectroscopy are in syllabus, and we will discuss one by one in further lecture. Thank you.